I guess I'll dive right in first off here with just kind of what's going on with platelets and what their role is in the bloodstream. And so I really wanted to explain like how first off platelets are even synthesized. And so we have our precursor megakaryocytes, and they form these long extensions, and those are in the bone marrow or the lungs, and your platelets come off and are released into circulation. Once they enter the circulation, they're there for about 7 to 10 days. And this is kind of the perfect time in terms of drug loading, and that's attracted a lot of attention because it's long enough that it can reach a bunch of different areas, but not too long that you have to worry about accumulation. Once they're kind of circulating, their main role is to stop bleeding. And so if you have any sort of injury, whether it be a paper cut or severe trauma, platelets are the first cells that are on the scene. And once they're there, they release a bunch of different microparticles that contain proteins and different factors that will recruit more platelets to come in and other coagulation proteins in order to form a clot and really stop the bleeding. In addition to that role, platelets will also release cytokines and chemokines at the sites of injury and have a really important position in the inflammatory response of these sites of injury. And so that's kind of just a general overview of platelets in terms of their general functions, but what about in disease states? And so platelets and cancer are intricately linked, and this is really highlighted by the fact that thromboembolism, so this would be clots that form in the bloodstream, are very highly common in cancer patients, to the point where it's the second most common cause of death in cancer patients. And it's second only to the cancer itself. And so one of the main reasons because of that is due to this platelet cancer loop that occurs. And so this figure is really going to walk through this interaction that occurs. And so cancer cells will increase and recruit platelets to the sites of tumors. Once the platelets are there, they're then activated and they release microparticles. These microparticles in turn will promote metastasis, tumor growth, and angiogenesis. And then that feeds forward and recruits more platelets, and you end up cycling between the cancer cells and the platelets themselves. In addition to that kind of interaction between each other, the platelets can also bind to the cancer cells and shield them from interacting with the immune system. And so you end up with cancer cells that are then able to move through the bloodstream and further spread metastasis. And so with that in mind, it seems very counterintuitive that you would actually end up using platelet transfusions in cancer patients, but they are frequently done. And this is because of the role that cancer treatment has on platelets. So chemotherapy and radiotherapy deplete the platelets that are present, and this in turn puts the patient at risk for bleeding, which is especially prominent in instances where any sort of surgery has to be done. And so cancer patients actually receive most of the platelet transfusions that are done clinically. However, they're not very effective at stopping bleeding. And there's also platelet refractoriness that can occur. And so this is when you repeatedly give platelet transfusions, but they don't actually increase the level of platelets that are in circulation. And this can happen in 7 to 34% of cancer patients that are treated with chemotherapy. In addition to that, there's also the risk of metastasis, which is explained through the platelet cancer loop. And so we want to overcome these issues by creating modified platelets. And so how can we control the function of platelets? And as with any cells, one way to control function is through the proteins that are synthesized. And so platelets, contrary to popular belief, can synthesize proteins. This kind of misnomer is usually attributed to the fact that they don't have DNA. And this doesn't really affect the proteins that are synthesized, but more the control. And so you can't target genetic methods or the DNA because there is no DNA. So you end up having to look at the RNA itself. And so for that, you really have to look at the key stages of translation. And so this would be initiation, where you have your ribosome binding to the mRNA to start protein synthesis, elongation, so it moving along your RNA and synthesizing your protein, and termination, so reading the stop codon and having your protein come off, and then your ribosome recycling so that it can synthesize more proteins. These 4 stages are really where RNA can play a big role in what proteins are synthesized and how you can control that. But it's not just messenger RNA, that's really where the control can happen. You have a bunch of other different forms of RNA that are also present in platelets. And so in addition to messenger RNA, your precursor pre-mRNA is also present in platelets, and a full spliceosome is also present. So you can have different isoforms of messenger RNA present, and that can control protein synthesis on one level. You also have microRNAs, which are small nucleotides that can really control which messenger RNA are present in circulation. These in turn are also really important in terms of bio-screening because there's different levels of microRNA in platelets depending on the disease state. And you can also, it's a really accessible kind of tool in terms of being able to take platelets out of circulation for that. Other RNA that's present, such as circular RNA and long non-coding RNA, are not as well studied, and so their exact role in terms of protein synthesis and controlling that is not as clear-cut. So now that we really clarified that RNA is one of the key ways to control protein synthesis and in turn platelet function, how can we modify the RNA? And so this kind of brings us into the fun part of the presentation, which is the nanomedicine that we've all been waiting for. And so with recent advances in the past several decades, there's been several different methods to try and deliver RNA to cells. And so several groups have successfully delivered microRNA and siRNA to platelets using electroporation and lipofectamine. And so these techniques are great in terms of delivering these small nucleotides because they're relatively stable. However, they're not as effective for messenger RNA. In addition to that, platelets are very easily activated, which is great if you're bleeding, but not so great if you're working with them on the bench. And so you end up with clots and other things forming in the tube, which is not really useful if you want to use this as a transfusible product. So lipid nanoparticles, or LNPs, have been really the forefront of the new technology in terms of delivering mRNA to platelets. And so this has been done by our lab, and this was research done by Stephanie Nowakowski. And so for this, she took platelets that had been labeled and delivered different types of LNPs that contained biotin labeled RNA in green. And so these are just some representative confocal images of lipofectamine, non-cationic LNPs, and just free RNA compared to cationic and ionizable cationic LNPs. And so you can see here that there's actually uptake of the RNA within the platelets in comparison to these other methods. And to just kind of give you a reference for what that actually looks like, this is the relative intensity of these confocal images. So you have this much higher uptake using cationic LNPs. And that makes sense because of the charge difference between the cationic LNPs and of course the RNA. Upon delivering these RNAs, we did not see translation in the platelets. And so that's something that we're really working forward and trying to optimize the LNP and also the RNA. And so before I wrap up here, I just wanted to acknowledge everybody in both the Kastrup and Cullis labs, and of course, all of our funding partners, which I would not have been able to do this research without. And I hope from this talk that you have seen the importance of the many different kinds of RNA that are in platelets and the key role this RNA plays in controlling platelet function. And so my work and those of others in the lab have really focused on improving the translation. And we've had some recent successes with that. So this talk is really the teaser trailer for the manuscript that we hope to have out later this year. So if you're interested, stay tuned. Additionally, we're constantly looking for new targets. So if anybody has any suggestions for anything that they'd want to put in a platelet or think would be interesting, we'd love to hear them. And so with that, I'd like to thank you all for listening, and I'd be happy to take any questions.
I'm curious, do you know how the platelets take up the LNPs?
Yeah, so there's a fun thing with platelets is the fact that they genuinely like to take things up in circulation. But the specific mechanism we had to go through and try in different knockdowns and things like that. And so using those kind of methods, we figured out that it's mainly through endocytosis. Past that, if we change the LNP composition, it might be a different thing, but for what we're working with that, it's through endocytosis.
Great, thank you. And then what can you see are the opportunities for using platelets in other diseases?
So one of the main things that we want to do is try and make the platelets more coagulable. And so in terms of cancer, that would be trying to deliver fewer platelets to them, but having the same results. But this would also be very useful in instances of trauma or severe hemorrhage, because then you could deliver fewer amounts of platelets, but also have the same effect in preventing bleeding.
Great, thank you. Marshall, are there any questions at this time?
No questions from the audience. Oh, wait, as I said that, one appeared. Nicholas Bertrand has a question, which is, it's written, so I'll read it out. What is the lifespan of platelets in vivo? If successful, how often would such a treatment need to be used?
So that's a really good question. Platelets, unfortunately, when they're collected to be used for transfusion, have a really short lifespan, and this is because they have to be stored at room temperature. So they're only able to keep them for about 5 days before they have to get rid of them and then collect more for more transfusions. And so you end up having to constantly change the platelets. And so with that, if successful, and we end up having platelets that are possibly more coagulable, it would hopefully decrease the number of transfusions that have to be done. But in terms of storage of the platelets in general, that they have a really short lifespan.